Welcome and thanks for joining for the presentation of SURFSara of the High Performance Machine Learning Research Team, where we conduct research in the field of histopathology. Today we will present to you our poster about Gigapixel semantic segmentation of whole slide images. All these whole slide images are slides of biological tissue that are extracted in the case of cancer to investigate the spread or the presence of metastasis in human tissue. Our work is connected to the Horizon 2020 ExoMode project, a European project aimed at developing exascale analytic tools for histopathology. Well, as the average pathology lab receives around 30,000 cases a year, this can be heavily supported by tools leveraging image recognition techniques, of which scalable methods need to be developed to deal with the exascale data coming from images and the patient reports. Our task in this research was the semantic segmentation of cancer tissue, and thereby also the classification on a slide level of the amount of metastasis present in the tissue. On this research, we focused on the data coming from the CAMELYON intelligence, which are whole slide images from the dataset of Radboud University in Nijmegen, Netherlands, which are common to research in the computer-aided diagnosis of tumor tissue. Well, the main challenge, as you see with these whole slide images, is the memory footprint, as these images can be approximately 200,000 by 100,000 pixels, which lead to considerable memory utilization at train time. At this point, this also means that the conventional training of the convolutional neural networks is not possible in data parallel settings, as even a batch size of 1 exceeds commonly used GPU RAM, which adds up on the whole slide image size and the activation maps. Another challenge considering the analysis of these images is the fact that different medical centers have different ways of color staining. This color staining consists of hematoxylin and eosin staining to visualize the cell nuclei and make them stand out of the cell plasma and surrounding tissue. However, the color distribution, as you can see, differs from slide to slide, and this can lead to inaccurate analysis of whole slide images. So what we want to emphasize here, what's also on the poster, is that the point made in introduction, where the whole slide images is high memory usage, is also shown. Because here we make an extrapolation of the memory consumption in one forward pass. And here you can see clearly that there exists an exponential relation between the patch size and the total memory. Also, we see that indeed we need to use CPU-based training, since the maximum memory of a common GPU, 32 gigabytes, indicated in the slide, is easily exceeded looking at the patch size. So in this poster, we showed that a larger field of view leads to higher performance of the semantic segmentation model. And this is done by looking at performance metrics like mean intersection over union. So the use of multi-scale delated convolutions means we can incorporate more information moving to a higher patch size. So this is shown here. We clearly see that there exists a trade-off between the accuracy and the memory consumption, as enlarging the field of view increases the mean intersection over union. This is not only across the field of view of one downsampling level, but also you see an increase of performance across downsampling levels. So indeed, a larger field of view leads to higher performance. This was enabled by CPU training on the Endeavour supercomputer by incorporating Horvath framework for distributed training. This allowed us to scale to 256 nodes with weekly scaling. However, even this constrained us to data parallelism on Intel Cascade Lakes with 192 gigabytes of DDR4. And this means we have to look further than data parallel methods. To conclude our poster, we see that patch size and zoom levels and the varying amount of information associated with it can be translated to a memory footprint, and this can be traded for accuracy. Medical imaging from high quality sensing equipment is a valuable digital source that could be leveraged with machine learning. However, to do this, we must do more scale-out experiments to even larger batch sizes with not only data parallel applications, but also model parallel applications. And we see also that in this case, then we can compare with compressed whole slide image analysis, which is valuable because then you can move forward to a gigapixel whole slide image analysis. Thank you for watching, and any questions, please contact me at the email address in the slide. Thank you very much.